All right. Well, thank you all for joining us this evening. My name is Jim Lockhart. I'm the president of Wild Connections, and we're happy to have you here for our indoor outing, Colorado's Cutthroat Trout, exploring the heritage of our state fish. Just to give you a little background, Wild Connections, now in its 26th year, is a science-based conservation organization which works to identify, protect, and restore wildlands in the upper Arkansas and South Platte watersheds, from the mountains to the prairies, to ensure the survival of native species and maintain the ecological richness of our region. Our goal is to protect not only wilderness and other undisturbed core areas, but also the corridors or landscape links through which wildlife can migrate. And we do this by a combination of science-based advocacy, collaboration with public land agencies, and on-the-ground restoration work. This indoor outing is one of a series which we've been presenting during the coronavirus crisis to keep people informed about activities in our state. And I'm very pleased tonight to introduce our presenter, Doug Krieger. Doug is originally from Michigan, where he received a BS degree in aquatic science from Michigan State University. And upon coming to Colorado in 1977, he attended Colorado State University, where he received an MS degree in fishery and wildlife biology. Doug has worked for the Colorado Division of Wildlife, which is now the Division of Parks and Wildlife, for 37 years as a researcher and area fishery biologist in northeastern Colorado, followed by a lengthy 25-year period as senior aquatic biologist, managing the aquatic resources within the Arkansas River Basin. Doug retired in 2018 after serving as Chief of Fisheries for the last 4 years of his career. So again, we thank you, Doug, for joining us tonight, and we appreciate the opportunity to hear from you about our state fish. So take it away.
All right, well, thank you, Jim. I appreciate that. I want to thank my friend John Stansfield for the invitation tonight. I really appreciate what this group does, and it's a pleasure to be with you tonight. We'll do this informal, and so if you have questions as I move along, be sure to get on that chat and move those questions to me, and I'll do my best to answer that. So with that, Allison, if I can share my screen. Okay, does everybody see that okay? Hello.
Looks good, thank you.
Okay, good. Very good. Many of you may be aware of how our understanding of Colorado trout has kind of evolved since 2012. But the science and the process of how we reach those conclusions is a lesser known, but still a really interesting story. So that's the one I'd like to share with you tonight, and kind of go through what that scientific inquiry looked like. So cutthroat trout are found in some of the most pristine montane habitats in the western United States. So it's no wonder why they are respected as an integral component of wilderness areas, and that conservation and recovery efforts have been undertaken to make sure that they maintain themselves in those landscapes. These fish require cold, clear waters, and they have been reduced in numbers over the last 100 years throughout the western United States by loss of habitat, overfishing, but most importantly, due to invasion of non-native trout like rainbow, brown, and brook trout. I'd like to widen out the view from Colorado and kind of look at the western United States. So there's somewhere between 12 and 14 subspecies. These aren't separate species, but subspecies of cutthroat trout throughout the West. Some of that's still debatable, and the science keeps evolving, like we'll talk about tonight. It should be noted that all these cutthroats are descendants of coastal cutthroat found in the northwest part of the United States along the Pacific coast. During a period of about what they understand is about 4 million years, the cutthroat have moved inland. So this period is has a lot of geological action. So there's been uprising of mountain ranges. There's been floods, glaciation. So what's happened has been this kind of hopscotch affair where uplifting of land masses has cut off the upper reaches of some of the streams and moved them basically into a new drainage. And that then they got populated. They were there for a while and became, evolved to that habitat and became kind of a special, their own subspecies. So that continued hopscotching from Yellowstone to Lahontan to Bonneville. And then it looked like our Colorado cutthroats, was probably evolved through about 1 million to 1.5 million years ago, transferred over into Colorado. So that's kind of the big picture. Human nature is such that we try to put labels on everything, and nonetheless, those are basically artificial. We try to use science to define species, subspecies, strains. But again, the fish really don't know those boundaries. And so I think the best thing that we've done is to heed the warning from Leopold. And the quote would be, to keep every cog and wheel is the first precaution of intelligent tinkering. And so that's kind of set the tone for trying to keep all these subspecies, and sometimes we don't even know what those are, keep those on the landscape as much as we can. So let's drill into Colorado. So this is a picture again of our understanding in the, back in year 2000, let's say, for instance. We have the Colorado River Cutthroat west of the Continental Divide. We have the Rio Grande Cutthroat down south in the Rio Grande drainage. And then the assumption that the greenback cutthroat trout was native to the South Platte and Arkansas drainages. That was our understanding. The yellowfin mentioned here was essentially found in just Twin Lakes up by Leadville, and by 1904 was determined to be extinct, largely because of the large amount of stocking in Twin Lakes during the heydays of Leadville silver mining, and that was basically found extinct by 1904. The Colorado River cutthroat and the Rio Grande cutthroat have both been challenged for listing under the ESA, but so far they've been able to stand those challenges, largely because there's a large number of populations and a fairly active recovery efforts there. But how we originally understood the distribution of what we assumed was the case in Colorado? Well, largely it was work done using morpheristic characteristics. So these are things, the physical characteristics of the fish, spotting patterns, the number of scales along the lateral line, the number of pyloric cecae, which is a little internal organ around near the stomach, the number of basiobranchial teeth and gill rakers. Those sorts of things were used to try to separate out. And it worked to a certain extent, but there was a great amount of variability. in those measurements and those physical characteristics. However, with trying to do conservation and recovery of these species, it's really essential to kind of know what their native range was, where they were actually endemic. And so with the advent of genetic work, the hope was that we would be able to better understand that. So as you know, the Human Genome Project got started in 1990. And by the year 2000, that science started, getting kicked down to things like cutthroat trout. So by the early 2000s, we were setting up contracts with a number of universities, BYU, University of Montana, Colorado State University, and CU, to take a look at some of those genetic capabilities that we could apply to cutthroats. It worked to a certain extent. The good part was that we were able to identify what we would consider pure Colorado cutthroats. So these were cutthroats that weren't hybridized with rainbow or Snake River cutthroat or Yellowstone cutthroat. So that was really a good quantum leap at that time, to be able to just make sure that when we had a cutthroat population, it was one of our Colorado cutthroats. But the tests weren't sensitive enough to differentiate between the Colorado subspecies, largely because our subspecies had not had evolutionary time to really have some really firm genetics that would differentiate it from some of the other subspecies that had been evolved hundreds of thousands or millions of years ago. Then we moved forward with the recovery team, and we contracted out a study by at CU, and it was completed by a PhD student, Jessica Metcalfe, an amazing researcher that took on a great challenge of trying to determine where our Colorado cutthroats actually were from. And so she used some new genetic techniques using mitochondrial DNA microsatellites to come to some excellent conclusions, and was able to finally differentiate between our Colorado cutthroats. So there was a long time coming. It was good news. This was information that had been desired by everybody involved in recovery of the greenback, and so it was much anticipated, and should have been really an occasion for dancing in the streets by those involved with the recovery. And although the advancement of the understanding was relished, there was some bad news out of this. And in fact, for those that have been like myself involved in recovery for 20 years, this genetic revelation was devastating. And I'll show you why. Here's a diagram from Jessica's paper in molecular ecology. And so she looked at a number of populations that are shown here. She had good fish representatives from many drainages in the state. But particular interest were the so-called greenback populations, these 9 populations in the Arkansas and South Platte drainages. Well, once she figured out being able to differentiate, it turned out that 5 of those 9 were actually Colorado River cutthroats rather than greenback cutthroats. So anyway, that was the tough news. Sometimes science advances. Sometimes it's good news, and in this case it was good news, bad news, because a lot of us had been involved that long. And all of a sudden, all this work that we had done, some of these populations were actually, you know, recovering Colorado River cutthroats. Well, CU had put out a news release, not probably a very well written news release, but the media picked up on it. And what the cause of the concern was that we were close to recovery on greenbacks. In fact, just before Jessica's paper came out in 2007, we were actually looking at delisting the species. We had exceeded the recovery goals in the greenback recovery plan, and we were moving along those lines. So a lot of money, a lot of time, but when this news came out to the media, they had a field day with it. So it was the so-called scientific blunder of all these dumb biologists have been recovering the wrong fish for decades and spending lots of money. So there was some heat that we had to take, but of course that's the media, and that's the price you pay, I guess, for advancing science. Going back and looking again at the conclusions that Jessica came up with in her paper was that obviously there had been some stalking going on, and this is stalking that occurred much earlier than any of us involved in recovery were aware of, and so that was troublesome. And particularly, here's this so-called greenback fish over in Antelope Creek within the Gunnison range. So what was all that about? Enter Chris Kennedy, who is a biologist with the U.S. Fish and Wildlife Service and kind of a closet fish genealogist. And he took it upon himself to start investigating stocking records to try to figure out when in history some of these things happened, because it was sure earlier than any of us involved had been aware of. So he started looking at records. He started, with Division of Wildlife stocking records that were some electronic, some in card catalogs. He went to hatcheries and attics and, dusted off, crates of old physical paper records. He went to the Leadville National Fish Hatchery, which was one of the oldest hatcheries east of the Mississippi, and looked at their records for decades and decades. Ended up actually going to the U.S. Fish and Wildlife Service repository that they keep for all their stocking records across the United States in Spearfish, South Dakota. Then he also looked at a lot of newspaper accounts. A lot of this stuff was captured. Stocking fish was kind of news back in the old days, and a lot of it got carried into newspapers. So after about a year of investigation, Chris found that there was a couple of big operations going on in our historic past. One at Trappers Lake, which many of you are aware of, east of Meeker and north of Glenwood Springs, and holding fish that represented the Colorado River cutthroats. And then Alexander Lake was on the Grand Mesa, and a lot of spawning going on there too, but fish that appeared to be this so-called greenback. Well, he came up with lots of records and lots of details, and one of them was how many fish had been moved around. So, basically from the Grand Mesa, 29 million fish were spawned and relocated, transported from the 10 years from 1899 to 1909. And then from Trappers and Marvine Lake, which was close by, 26 million fish at the beginning of the 20th century. it's maybe a little hard to see these lines, but stocking records show that these fish were all distributed to basically every county in the state that had suitable habitat, cold water habitat. So lo and behold, that was quite the revelation that many fish had been moved so early in our history. So what that did was really confuse the situation in terms of what was endemic. Where did these fish originally come from? And obviously, all the landscape of fish got the whole puzzle pieces were pretty well scattered by 1900. So the idea was, do we have any representatives of fish that were maybe collected back in history before 1900? Well, when you get confused, you go see the guys in the white coats. And in this case, the white coats were curators of museums, and in some of our foremost museums across the United States. We were aware that those samples existed. We didn't know how many, we didn't know exactly the location of them, but we were curious to find out if that held any possibilities of helping us determine where these fish might have been early in the state's history. So we, again, the recovery team contracted with CU Boulder, and with a idea for a study to compare the genetic composition of the cutthroats that are currently existing in Colorado to those historic museum specimens that were collected again before 1900. And in that study, to describe and trying to determine the geographic origin, the evolution, and what kind of distinction there were between the cutthroats in Colorado. Luckily, we had some, outstanding early expeditions, well funded for the time, and headed up by a lot of great naturalists that took excellent care of collecting and curating lots of invertebrate and plant species, and putting them into museums. So, Hayden, Ferdinand Hayden, you're aware of. David Starr Jordan was a notable collector of the time. You have Edward Cope, and another one that may be familiar to some of you locally here is Charles Aiken, who came to Colorado Springs in 1871, and he also did some expeditions with the Wheeler expedition in Southwest Colorado in 1874. So let's take a look at those collections. Again, there was not very many places to take those fish, but there are renowned collections that have gone through the test of time. So we've got Smithsonian, the California Academy of Sciences, Harvard, and the Academy of Natural Sciences in Philadelphia, where all these samples were archived many, many years ago. So that took a while to figure out where all those fish were. The other good part of the story was that Jessica Metcalfe, who had done the work in 2007, was able to come back, and she was interested in continuing this saga. She had, in the meantime, between 2007 and 2012, had worked at the Australian Center for Ancient DNA, where she got tremendous experience. They were working on things like mammoth that had been gotten out of old glaciers. So she came back and had the techniques to be able to recreate some of these mitochondrial DNA segments, patch them back together, which is a very difficult laboratory function, and primarily looking at this ND2 gene on the mitochondrial DNA, which is real diagnostic, is the real diagnostic gene for cutthroat trout and basically in almost all trout. So again, what you wanted to do was to compare those genetic fingerprints of those museum specimens that we were housed away to fish that were currently found in Colorado at the time. So here's the samples that were available. The drainage, almost all the drainages in Colorado, luckily were sampled by early explorers. Here's the localities were the number of different collections. So the most collections were in the South Platte drainage, where there was 5 separate collections, and some were just one collection, and some, like the San Juan here, were only 2 fish. I might also refer you back just a second again to that San Juan sample. Just remember that's Charles Aiken's work, where he had 2 fish stored away in the Smithsonian from the San Juan as well. So anyway, let's fast forward then to her results. There's lots of methods and lots of details we won't go into tonight, but we'll just jump to the conclusions. So first of all, let's take a look at, again, here's what our situation looked like with the assumptions of Colorado River cutthroats to the west, greenbacks in the South Platte and Arkansas, Rio Grande's down here, and yellowfin's extinct. The next slide, what we're going to do is get away from actual names. Names, as we move forward, become confusing, actually. And it's better to do kind of a Dr. Seuss approach of looking at colors. So we've got green fish, we've got blue fish, we got yellowfish, we got orange fish. And so, again, the green is the fish formerly known as greenbacks. The blue are fish formerly known as Colorado River cutthroats. The orange fish are the Rio Grandes, and the yellows are the yellowfin. So now I'm going to show you, this is the pre-study. Now I'm going to show you post-study. So here again, quite some changes. Most notably, I'm going to go back and forth here to let you take a look at what's happened with this information. Historically, we thought we had the 4 subspecies of cutthroat trout in Colorado. With the advent of this research, we found indeed that we had 6 original subspecies in Colorado. So with the addition of a South Platte cutthroat and a San Juan cutthroat. The rest of the fish, we had representatives on the landscape, or at least collections like the yellowfin. So let me go back and forth again. So the green fish, thought to be in the South Platte in Arkansas, formerly known as greenback, restricted to the upper Colorado River drainage, as well as the Gunnison and Dolores, based on those historical samples that were out there. The blue fish, the fish formerly known as the Colorado River cutthroat, now was restricted and endemic, apparently, to the lower Colorado River drainage. The Rio Grande, of course, didn't change any, but it also was thought that, and according to some of Chris Kennedy's work, looking at newspaper accounts, that the yellowfin cutthroat was probably not just restricted to Twin Lakes. There was very little sampling done in the Arkansas, unfortunately, so we don't have historic samples taken like at Salida or Buena Vista or Canyon City, so we don't know that for sure, but it's likely that the yellowfin was endemic to the Arkansas. So what do we know then? Sorry about that. So compared to the museum specimens that helped us locate where the endemic range actually was before all the stocking occurred in the state, we see that the yellowfin, we don't have any representatives currently on the landscape that match the genetics of the yellowfin. That's assumed to be extinct. At the time, we also didn't have any San Juan River cutthroats, no current genetics to compare to those two fish that were collected by Charles Aiken back in 1874. But how about the South Platte River? Well, turns out that there was one population of fish on the landscape in Colorado that matched the genetics of those museum specimens. And again, there was 5 collections, quite a number of fish collected in the South Platte River Basin very early. And so here's the fish, the South Platte cutthroat that I'm holding. And many of you probably know that that fish actually came from Bear Creek, small drainage just west of Colorado Springs. So the the fish in Bear Creek are restricted to 4 miles of habitat. Not a lot of water in that drainage, but good, clean, cold water coming off of Pikes Peak. It was separated from down below by a natural barrier shown right here that allowed those fish to be isolated for apparently eons. So the question would be, how did these South Platte fish that were native to the South Platte, how did they get into Bear Creek, which is actually in the Arkansas drainage? Luckily, the information that Chris Kennedy got into helped us discover what exactly happened. And that was through largely newspaper accounts in the Colorado Gazette-Telegraph, which it was called at the time, and found out the history of these fish. So it turned out that the U.S. Signal Corp was trying to build a trail along Bear Creek to the summit of Pikes Peak where they wanted to set up a station. They looked at a number of routes. This one was a longer route, but a better route than other ones they could determine. But this this route was back in those days was 18 miles, was a 2-day trip. Now it turns out that a gentleman named of Joseph C. Jones homesteaded 160 acres along Bear Creek in 1874 with the idea that he wanted to establish some kind of inn. He knew that it was going to take a 2-day trip. He wanted to make some money, and that was his plan. Also, there was another publication, a book that was published a long time ago in 1882, excuse me, that had a quote from a Reverend Cross that wrote the book, that Jones strode back to the work of digging stones out of his fish pond. Historical records show that largely Fountain Creek drainages had been devoid of fish, and so Jones must have brought those fish in from somewhere. The most likely source of any kind of cold water fish at that time would have been Trout Creek, which is in the South Platte drainage that flows, north out of Woodland Park towards the South Platte and drains into the South Platte near Deckers. So most likely that's how the South Platte fish got into Bear Creek, and the barrier allowed those fish to be isolated, since they were probably stocked back in 1980 or 1880 or so. So then Bear Creek became quite important. In fact, by 2008, after Jessica's first publication, 2007, she had noted that of the fish that she looked at, the Bear Creek fish were somehow unusual. And we recognize that as well. They just physically have different spotting patterns and a look than in the rest of our cutthroats that we had on the landscape in Colorado. So we started protecting it early. 2008, we started doing some work. We formed a Bear Creek team, and actually a group of involved citizens, interest groups. And Jim Lockhart was one of the founding members of that group, representing Sierra Club. We got to work and doing things like bridge crossings, alleviating all the sedimentation, which is the risk to Bear Creek. Of course, it's Pikes Peak granite that erodes very easily and threatens the habitat for this fish. So that was of great concern. A lot of work done by Rocky Mountain Field Institute on restoration of trails up there. There's been changes in use, changes of cutting out motorized use in some areas, and just most recently, El Paso County, which now holds the what is called Jones Park after Mr. Jones. They now manage that, which is the heart of Bear Creek and the lion's share of the habitat for the fish. And so they put out a master plan, and if that master plan is followed, we have a really good opportunity to enhance this fish and its habitat to make sure it stays secure. So good work by lots of folks. So where do we go from here? Like in 2008, we actually took horses in and removed about 65 fish, moved those fish into a couple of hatcheries, Leadville National Hatchery and our CPW Mount Shabana Hatchery at Salida, where a brood stock was established, protect those fish and to produce more young fish for translocation to the South Platte drainage. One of the first, actually the first reintroduction of this greenback. It's actually referred to as a green lineage fish because the name greenback is not sure if that'll stick with this fish or not. But it was stocked in Zimmerman Lake up by Cameron Pass in the upper Poudre drainage. Those fish then became established, and we started spawning those fish in 2018 to add more wild fish to the mix of fish that were being also produced at the hatcheries. So we've been after that for a number of years now. Those fish are usually planted as small fingerling fish, packed up in bags with water and oxygen, and basically backpacked in by volunteers to different locations. Here's a list of translocation projects that have been already started and our future projects coming up. Zimmerman Lake, like I mentioned, was the first one. There's a couple here that are in the Clear Creek drainage, Herman Gulch, Dry Gulch, West Fork of Clear Creek. There's some in South Park, like Rock Creek and Black Canyon Creek. There's also some more candidate waters in the upper Poudre, again, in habitat that was formerly used for recovery for greenbacks. Those fish are going to be removed, and the now true green lineage Bear Creek fish are going to be placed in there. Another huge project is proposed for the very headwaters of the Poudre drainage that also goes into Rocky Mountain, the north side of Rocky Mountain National Park, where there's potential for 37 miles of habitat and 242 acres of standing water. That's a huge project that's slated to begin in part by 2022, but it'll probably take over a decade for that one to really get established. One thing I'd like to jump to just to let you know was that when Jessica finished her work in 2012, we didn't have any fish that looked like the historic samples on the San Juan River. My prediction at the time was that we would find some. There hadn't been many genetic studies done in the San Juan River drainage. A lot of that country is remote, and so there wasn't actually a lot of fish samples taken. But after Jessica's work, the push came on to start looking at a lot of fish down here. And come to bear was that in 2018, we found 2 populations of fish, tested those genetics, and those matched up with those 2 fish that were collected by Charles Aiken in 1871. So again, now we have the South Platte fish on the landscape, as well as the San Juan River fish. So the only fish that's still lacking is the yellowfin. I doubt that will be found. We've done a tremendous amount of work in the Arkansas, looking at almost every stream there is and subjecting that to genetic tests. So it's unlikely, not impossible, but unfortunately, the Arkansas is the one fish without a endemic native cutthroat to it, unfortunately. One other side note, it's interesting that just yesterday, a discovery that a giant tortoise species was found based on the genetic work, just like we talked about with the cutthroats tonight. A female found in 2019 and genetics were taken that match up to a sample that was taken on a tortoise back in 1906, and they matched. And so this species was apparently considered extinct, and now with the genetic findings, actually was established now as a remnant of that once lost population. So science keeps evolving. We haven't seen the end of what genetics can do for us yet, but luckily we have a lot of specimens tucked away in lots of places that we can use to examine species that are on the landscape today. With that, I'll conclude, and I'll be glad to take any questions.
So,
thank you, Doug. You do have one question in the chat box, which you could answer. From Mary Joe Lewis, was the Zimmerman Lake project impacted by the Cameron Creek fire?
Good question. No, it wasn't. It was close, but didn't burn that location. Unfortunately, one of the populations I showed on that list was East Fork of Roaring Creek, and fish had just been put in, stocked in June, after many years of getting that habitat ready for fish. And so that was going to be another great location for these Bear Creek fish, but that burned heavily. And so that population was lost, unfortunately.
Okay, Doug, I do have a question about Bear Creek. You've said that they've moved some of the fish into hatcheries and they're now breeding stock in some other locations. Could you explain a little bit about why it's still important to have these wild fish in their so-called native habitat as opposed to just raising them in hatcheries?
You bet. We've talked a lot about genetics of those fish, but compared to a lot of other cutthroats in Colorado, excuse me, compared to a lot of cutthroats in Colorado, the genetic, basically library that contained in that fish population is fairly limited. For one thing, we don't know how many fish Mr. Jones actually moved to establish that population, but obviously, he took some fish with certain genetics to form that population. Now we took some fish out, 65 fish, to start a brood stock program. But the lion's share of the genetics, all that we have in the whole world, is contained in Bear Creek. And so that's still the mother population and still is vital. There's going to be work done this year trying to get more genetics. Those fish have been spawned every year, and you try to capture some of that wild information, that those genetics, to add to the populations in the hatchery to when you're producing these fish. So those are those are all the fish we have in the world for Bear Creek, and that's why it's so important to maintain that population. Why that became so important to CPW, the U.S. Fish and Wildlife Service, the Forest Service, and now El Paso County. We have seen, excuse me, the CPW biologist did see quite a decline in fish last year, fairly significant decline, in fact. But it was also found in places like Sudbury and North Catamount Creek. So there seemed to be something going on physically, biologically, that reduced the numbers of fish. Now, obviously, the fish have been there for 135 years. They've gone through lots of different, probably, cycles, and including road building through Bear Creek. The seven lakes were built on the south slope. All the material, all the concrete, all the materials, all the metals had to came up a road through Bear Creek. So these fish have withstood a lot, and it's hopeful that they'll keep maintaining for another 135, 140 years.
There's a question from Bob Ballard asking, when will Rock Creek and the Lost Creek Wilderness receive fish from Bear Creek?
They're still working on that project. There's a lot of these projects are done in segments, and there's you have to remove the non-native fish. You have to build barriers so the native cutthroats are not invaded by non-natives that would either reproduce and hybridize or outcompete those fish. So those are all pieces that have to be put together. Also, in some of these locations, there's some remnants of whirling disease. What has to be done is to break the cycle of whirling disease by taking out all the trout for a 3-year period. That breaks the life cycle of this parasite. And after that time, then the whirling disease no longer becomes an issue, and you can go forward with those reintroduction efforts. So it's on Rock Creek's on the list, and work has already been done and started, but it'll be a few more years yet.
Okay, we have another question. What is being done to investigate why the populations were declining this last year?
The biologists have been investigating a number of aspects, primarily with temperature. So there's lots of temperature thermistors, recording thermistors that log temperatures every 15 minutes. So there's a number of those throughout the whole Bear Creek drainage. So looked at temperatures. We have good flow records because there's a gauging station right at the bottom of Bear Creek. Looked at air temperatures and all sorts of factors. Looked at try to figure out if there been any significant predation, if there was any kind of illegal harvest of fish going on, if had been any contaminants that might have been put into the water. But none of those have really resulted in any kind of answers yet. So the biologists are still kind of scratching their heads. It's just a hint that the other nearby drainages also have been sampled and also show that same kind of decline. So it looks like it may have been just a kind of a sudden situation with perhaps flows and temperatures that might have been really tough on fish for a fairly kind of a short period of time. They've talked to people that hike up there regularly, and nobody have noticed any kind of fish kill. But sometimes you may not see that.
Okay,
Still work to be done there.
let's take one more question and then we'll wrap up. And that is, are there plans in place to mitigate cutthroat habitats from the changes from climate change impacts?
Yeah, there's lots of initiatives occurring right now at the state and federal levels in terms of trying to do everything that we can to maintain the habitats as they are. So that involves making sure that we maintain good forest, good riparians. And so those are key. Those headwater situations are really key for this. There's, since we're working on recovery at headwater streams, some warming of that water actually would be beneficial to the fish. But if that if those global changes, climate changes occur along with a reduction in flows or hot daytime temperatures, then that would not help the situation at all. But lots of initiatives there. One of the main things you can do with cutthroats is to make sure they're into a lot of different habitats across the state. The more places you have, the more redundancy, the more security you have in those fish. So that's one of the prime things that's is being done with all our native cutthroats.
Okay, let's wrap up with one more question. Is there a cutthroat trout called the greenback anymore?
So the U.S. Fish and Wildlife Service is working on putting together a species status assessment, what they call an SSA, which is required for the greenback. And that will be used as a basis to form a new recovery plan. There's still some question as to the actual label for these fish, if the greenback is actually the appropriate label. The type specimen, every species has a type specimen that's used for identification and locality and a name. But the actual fish that's called a greenback is actually turned out to be a Rio Grande cutthroat. And so we're not sure if that name is actually going to carry over to this new green lineage Bear Creek fish. That's yet to be determined.
Okay, well, thank you very much. That was very informative. Jim, do you want to wrap things up?
Well, yes, thank you very much, Doug. That was a great presentation. And as Allison said in the beginning, we are recording this, and we will make it available to people. If you have any of your friends who wanted to attend and weren't able to, we will be able to put it on our webpage, and you can look at it there. If you like to learn more about Wild Connections, you can go to our webpage. It's www.wildconnections.org, or go to our Facebook page, which is facebook.com/wildconnections.org. You can also join us on our hikes. And of course, you learn more about our activities, ways you can help the environment. And of course, we're always grateful for donations to help support our endeavors. So once again, Doug, thank you very much for the very informative presentation. I think we've had a great time on these indoor outings, and I expect we will continue them even now that the opportunity to get back into the outdoors is beginning to be upon us. So we hope to see you all in some of our future events, our future outings, our future indoor outings, and have a good night, everyone.
Okay, thank you, Jim, and thank you, everybody. Good questions and very attentive audience. I appreciate it. It's been a pleasure being here with you.